Welcome to this explainer. When we look at a beautiful flower, we usually just appreciate the vibrant colors and that pleasant fragrance. But today, we're going to completely reframe how you look at a blooming garden. We're going to examine sexual reproduction in flowering plants, but definitely not as some dry, static textbook process. Think of it more as an epic, microscopic journey of survival, adaptation, and just incredible biological engineering. Okay, let's dive into this with a quick three-part roadmap. First up, we'll look at the anatomy of pre-fertilization. Second, we'll cover the double fertilization process. And third, post-fertilization, focusing on seeds and fruits. So, kicking things off with Section 1, Anatomy of Pre-Fertilization. Before any of the real action happens, we first need to meet our primary structures, the male and female reproductive organs. To ground this anatomy in reality, take a look at the pistil of a hibiscus flower right here. This is the female reproductive organ. At the very top, you've got the stigma, and this acts as a highly specialized landing platform. If you've ever touched an opened anther of a hibiscus, you've probably had those yellowish, powdery greens rub off on your fingers, right? Well, that's the pollen. The stigma's entire job is to catch that pollen, kicking off a journey down that long, slender stalk called the style, until it eventually reaches the basal bulged part at the bottom, which is the ovary. Now, before pollen can even land on the stigma, these reproductive elements actually have to be created. Let's clear up some heavy scientific jargon here so you can see exactly how the male pollen and the female embryo sac are prepared. On the left, we've got microsporogenesis. This happens in the male stamen, specifically forming male microspores, which become pollen grains, through a cellular division called meiosis. Then over on the right is megasporogenesis. This takes place in the female pistil, forming female megaspores from a single mother cell inside the ovule. Basically, this is the biological prep work. Now, what's really interesting about this slide is the fascinating biological math happening inside the female gametophyte, also known as the embryo sac. It all develops from just a single functional megaspore. Its nucleus divides mitotically, but get this, it doesn't form cell walls right away. It keeps dividing until there are exactly 8 free nuclei just floating around in there. Only then do cell walls eventually start to form around most of them. The result? A highly structured, mature embryo sac that has 8 nuclei, but incredibly, only 7 distinct cells? And among these are the absolutely crucial egg cell waiting at one end and a large central cell holding 2 polar nuclei. Moving right along to Section 2, the double fertilization process. This is really the climax of our story, which begins after pollination finally brings the non-motile male and female gametes together. Since plant gametes can't just swim or move on their own, they need a sophisticated delivery system. Enter the pollen tube journey. Once a compatible pollen grain lands on the stigma, it germinates and literally grows a microscopic tube all the way down through the style. And this isn't just passive growth, it's an active, chemically guided mission. The tube navigates its way directly into the ovule, specifically entering a cell called the synergid, all to deliver 2 male gametes exactly where they need to be, to set the stage for the main event. Once those 2 male gametes are released, a perfectly choreographed, sequential 2-step process kicks off. Let's walk through it clearly. Step 1 is syngamy. One of the male gametes moves to the egg cell and fuses with its nucleus to form a diploid zygote. That zygote is what's going to eventually become the new plant embryo. Then we have Step 2, triple fusion. The second male gamete moves to the center of the sac and fuses with those 2 polar nuclei we mentioned earlier. Since this involves 3 haploid nuclei coming together, it forms a triploid nucleus. So the crucial point is this concept of double fertilization. This is an event entirely unique to flowering plants, the angiosperms. You have 2 completely distinct types of fusions happening simultaneously within a single embryo sac. Syngamy, creating the future plant, and triple fusion, creating a nutrient source. It is an absolute marvel of evolutionary timing and efficiency. And that brings us to our final act, Section 3, post-fertilization, focusing on seeds and fruits. Now we're going to explore the legacy of fertilization and how these microscopic events create the tangible parts of nature we actually interact with. Remember that triply nucleus formed during triple fusion? Well, it develops into the endosperm. This is a vital tissue packed with reserve food materials. Biologically, the primary endosperm cell rapidly divides to create this guaranteed food supply before the embryo even begins to really grow. It acts as a ready-to-eat meal designed specifically to nourish the developing plant embryo. To relate this dense biology back to a real-world example, let's look at the coconut. In a lot of plants, the endosperm is completely consumed by the growing embryo. But in others, like the coconut, we get a persistent endosperm. It survives in the mature seed to aid in future germination. This is kind of wild. When you crack open a coconut and drink the liquid water, you are actually consuming free nuclear endosperm. And that rich, white coconut meat, that is the cellular endosperm. You are quite literally eating the plant's biological reserve food supply. Meanwhile, what is the embryo up to? The diploid zygote brilliantly adapts by simply waiting. It enters a period of dormancy, waiting to divide until a certain, safe amount of that endosperm is formed. Think about the evolutionary brilliance of this pause. The plant prioritizes survival above all else, refusing to start growing the next generation until it has absolutely guaranteed the nutrition required for the embryo's success. As the embryo finally develops and matures, a grand, simultaneous physical transformation takes place. The mature ovules inside the flower transform into seeds. At the exact same time, the wall of the surrounding ovary thickens, expands, and develops into the fruit. So, a fruit is essentially just the protective, ripened aftermath of the ovary, perfectly designed by nature to shield those seeds and eventually help disperse them out into the world. And this brilliantly illustrates a really fun botanical distinction. We have true fruits, which develop exclusively from the ovary. Think of a guava or a mango. But then we have false fruits. In false fruits, other floral parts, specifically the thalamus at the base of the flower, swell up and actually contribute to the fruit's formation. Because of this structural involvement, an apple is biologically considered a false fruit. The crisp, fleshy part you eat isn't just the ovary, it's mostly the expanded thalamus. We've traced this incredible, intricate journey from microscopic spores all the way to the fruits sitting on our kitchen tables. To wrap up this explainer, I want to leave you, the focused learner, with a direct, thought-provoking question to ponder. Is there a relationship between the number of ovules in a flower's ovary and the number of seeds present in its fruit? Next time you slice open a single-seeded mango or a multi-seeded watermelon, take a really close look. See if you can deduce the microscopic history of the flower it came from. Keep observing, keep learning, and thanks so much for joining me.